Assalamualaikum students. Before going to our educational topic, please subscribe my channel Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is frame shift mutation. Frame shift mutation is the type of gene mutation. So before discussing the frame shift mutation, first we see the definition of gene mutation. A gene mutation is basically a change in the nucleotide sequence of our DNA molecule other than genetic recombination. In other words, we can say that a gene mutation is a change, a variation in the composition of the DNA sequence that commonly occurs during meiosis and mitosis. Gene mutation consists of two types. The first one is point mutation and the second is frame shift mutation. Point mutation, also known as base pair mutation or base pair substitution. I have already prepared a lecture for the topic point mutation, during which I have explained different types of point mutation. You must watch it for clarifying your concept about point mutation. That was the focus of the previous lecture. But in this lecture, we are going to focus on frame shift mutation. Frame shift mutations are the result of insertions or deletions that take place on our DNA molecule. Let's define what an insertion is and what a deletion is. An insertion basically refers to the addition of a nucleotide base pair or several nucleotide base pairs at some given location on our DNA molecule. On the other hand, deletion is the removal of a nucleotide base pair or several nucleotide base pairs at some given location and given position on our DNA molecule. Now, as we will see in just a moment, an insertion or deletion may or may not shift the reading frame of our messenger RNA molecule. Now, what exactly is the reading frame? Well, the reading frame is the sequence of nucleotides and the order of the nucleotides. In simple words, we can say that a reading frame is the sequence of codons that are present on the messenger RNA molecule. These codons are read by the ribosomes during the translation process. A translation process is the synthesis of polypeptide chain during which a cell synthesizes its required protein. I have also prepared a lecture for translation process that you can see easily at my channel Mehdi Science TV on YouTube, during which you can easily understand the mechanism of polypeptide chain synthesis. So let's take a look at an example of an insertion that causes the shift of our reading frame, and as well as see in just a moment that is known as a frame shift mutation. So let's begin by looking at our DNA antisense strand that is used during the process of transcription. You can also watch the transcription lecture in detail at our channel in English as well as in Urdu language. So let's suppose this is our DNA strand. The 5 end is here and the third end is here. So we have GCGAG CCTAG is the nucleotide sequence on our DNA antisense strand. Now when we actually transcribe our DNA molecule, then our DNA polymerase enzyme reads beginning on the third end the DNA and joining towards the 5 end. So that means when we synthesize the messenger RNA molecule, we have to look going this. So where the complement to G is C, the complement to A is U, the complement to T is A, the complement to T is G, and all others will be A double C to their complements. That will be U double G. So basically this is the 5 end of the messenger RNA and this is the third end of the messenger RNA molecule that is produced during the process of transcription. When we transcribe this DNA antisense strand, now once when we actually synthesize the messenger RNA molecule, we are going to use this messenger RNA molecule and the ribosomes which are going to translate the codons of messenger RNA molecule, their nucleotide sequence into the amino acid sequence, and the ribosomes will use the genetic codes which consists of 64 codons. So our codon CUA corresponds to the amino acid leucine, whereas the codon of GCU corresponds to the amino acid alanine, and the codon of CGC corresponds to the amino acid arginine. So let's suppose as a result of some type of outside factor, we have an insertion taking place. So we have a mutation that is known as insertion mutation. So basically, let's suppose between the 7th and the 8th nucleotide on the DNA antisense strand, we insert our cytosine nucleotide. So now our DNA strand consists of 10 nucleotides instead of 9. So we have the mutated DNA antisense strand. We have in this strand nucleotide as G, C, G, A, G, C, T, and inserted nucleotide cytosine, and then again A and G. Cytosine is present between the 8th and the 7th nucleotide, where this is the cytosine that has been inserted between the 7th and 8th DNA nucleotide. So let's see what kind of polypeptide sequence of amino acids were actually going to form. So when this molecule is transcribed, we form this mutated messenger RNA molecule. The reason for its mutation is that it contains this guanine between the 2nd and the 3rd nucleotide. So the guanine becomes a cytosine. The complement of A is U, the complement of E is A, the complement of C is G, the complement of G is C, the complement of A is U, and so forth. So this is the 5 end of the messenger RNA, and this is our 3rd end. Now when our ribosomes read this mutated messenger RNA strand, we'll use the genetic codes. But now because we have a shift in the reading frame, what will happen in the codons, that will change. So the first codon in this case is CUA, whereas the first codon in this case is CUG. But because CUA and CUG basically code for the same exact amino acid, leucine, first amino acid is not changed. But look what happens to the next because we insert the single nucleotide into our sequence. It shifts all the other nucleotides, that changes the codons that all read by the ribosomes. So in this case, the second codon was GCU because in this case we had a shift in the reading frame. The second codon is AGC, which is different than TCU codon. Then this one is CGC, but in this case the third codon is UCG. Now see, CUG still codes for leucine, but AGC codes for amino acid serine, and you can see the next codon UCG codes for serine amino acid next again. So these two codons codes for the same exact amino acid, and notice that this or this for that matter is different than the sequence. Even here we have serine and next we have alanine and arginine amino acids. So we see that the insertion of the cytosine nucleotide between the 7th and the 8th nucleotide of the DNA causes a shift in the reading frame of the messenger RNA molecule. And this insertion changes the codon that are read by the ribosomes, and this changes the sequence of amino acids that are produced on the polypeptide chain, and this result usually leads and produces a non-functional protein. So we see that any deletion or insertion of a nucleotide that is not a multiple of 3 nucleotides will cause such a mutation, such a shift in the reading frame, and such a mutation is commonly known as frame shift mutation. So we see that a frame shift mutation is caused when we have either an insertion or a deletion of nucleotide or nucleotides that are not a multiple of 3. But what exactly happens if the insertion or deletion occurs in a multiple of 3 nucleotides? So let's suppose and let's consider what happens when we insert 3 nucleotide sequence into the previous antisense strand, that is DNA antisense strand. Let's see what the end result will be. So let's begin with this DNA antisense strand. Once again we have the GCGA GCTAG nucleotide sequence from 5 to 3rd direction of DNA antisense strand. If we transcribe this, we produces the same messenger RNA strand that we have already prepared. When we translated them on the messenger RNA, we produces once again the amino acids leucine, alanine and arginine, that are the amino acid sequence. Now instead of inserting a single nucleotide which is not a multiple of 3, suppose we insert a multiple of 3 nucleotides. So we exactly insert a multiple of 3 nucleotides CAG nucleotide between our 6th and 7th nucleotides on the DNA antisense strand. So we basically enter the sequence of CAG. Now when we transcribe this, the mutated DNA antisense strand we produce, so the G becomes a C, the A becomes a U, the T becomes a, the G becomes a C, that A becomes a U, C becomes a G, again C becomes a G, the G becomes a C, the A becomes a U, and GCG become CGC nucleotide sequence. So this is our mutated messenger RNA strand, and when we actually use the ribosomes and the genetic code to translate this mutated messenger RNA strand, we produce the following results. So the CUA is the leucine that doesn't change. Now that we insert these 3 nucleotide sequence, this sequence CUG corresponds to the amino acid leucine. Now next we go on to the GCU, which is the amino acid alanine, and the CGC, which is our arginine. Now notice that what happened, there is a big difference between the previous result and this result. So because in this case we had a frame shift mutation, because we only inserted a single nucleotide, shifted the reading frame, and that changed all the amino acids that were produced after our mutation. But in this case what we essentially do, we don't actually shift the reading frame. So what we do is we simply insert a single amino acid, and in this case but all the amino acids that are produced after the inserted amino acids are exactly the same as before. So we have leucine, alanine and arginine, and also we have leucine, alanine and arginine. Basically the only difference between the polypeptide chain produced in the non-mutated strand case and in a mutated strand case is our single amino acid that is found between the second and the third amino acid. All the other amino acids and their positions are exactly the same and is exactly the same. So whenever an insertion or a deletion takes place in multiple of 3 nucleotides, this doesn't lead to a shift in the reading frame, and such mutations are known as non-frame shift mutations, because all the amino acids that would be produced if there was no mutation are still produced, and this usually leads to either a fully functional protein or a partially functional protein. And if we compare to the frame shift mutation that usually lead to a non-functional protein, so basically insertion or deletion do cause frame shift mutation, but not always. Sometimes they can cause non-frame shift mutations. Those basically means that don't shift the reading frame of the messenger RNA molecule, the reading frame that is used by our ribosome. What we basically do is we insert or delete one or more amino acids, but all the other amino acids basically remain exactly the same. So to conclude, we have two main types of mutations: point mutation, which are basically mutation on a single nucleotide, and these are also known as base pair mutations, base pair substitution. Now we also have frame shift mutation, and frame shift mutation are caused by insertions or deletions. But even though insertions or deletions cause frame shift mutations, sometimes they can also cause non-frame shift mutations. This was an introductory video about frame shift mutation, including insertion and deletion. In our subsequent videos, we will discuss about recombinant DNA technology, respiration, glycolysis, Krebs cycle, and many other topics. So make sure to subscribe our channel for further upcoming videos. Also make sure to like my videos at YouTube channel Muntazir Mehdi Science TV for receiving notification of next videos. At the end, thank you for watching this your YouTube channel.